The overall goal of this procedure is to isolate adult stem cells from musculoskeletal soft tissues, such as skeletal muscle. This is accomplished by first taking a biopsy of the soft tissue, debriding it, and chopping it finely. The second step of the procedure is to enzymatically digest the tissue. The third step of the procedure is to isolate the stem cells through repeated pre-plating steps. The final step of the procedure is the identification of the isolated stem cells. Ultimately, results can be obtained that show the cell's morphological and cellular behaviors using immunofluorescence microscopy and/or flow cytometry.
Hi, I'm Yun Li from the Laboratory of Molecular Pathology in the Department of Orthopedic Surgery at the University of Pittsburgh. Today, I will show you how to isolate adult stem cell from the soft tissues of a musculoskeletal system, such as skeletal muscle.
Hello, I'm Dr. Johnny Ward. I'm a professor at the University of Pittsburgh School of Medicine within the Department of Orthopedic Surgery. I've been working, closely with Dr. Young Lee on these procedures to isolate, different type of stem cells from skeletal muscle and other tissues such as tendon.
We use these procedures in our laboratory to isolate stem cell for basic research studies and translational medicine applications. So, let's get started.
The tissues from which stem cells can be isolated include tendons and skeletal muscles harvested from the tibia or soleus muscles of the hind limb of wild-type mice. After removing remnants of skin and bone, place tissues in a dish containing cold HBSS supplemented with 5% FBS. Next, place the tissues separately on collagen-coated dishes containing cold HBSS. We will use skeletal muscle tissues to demonstrate the basic procedures for tissue dissociation and digestion. Use microscissors and/or scalpel blades to mince the skeletal tissues into a coarse slurry. For enzymatic digestion of the tissues, start by transferring the minced tissue slurries into separate 15 milliliter tubes and centrifuge at approximately 3,500 RPM at 4 degrees Celsius for 5 minutes. Remove the supernatant, wash by resuspending in HBSS, and repeat the centrifugation. After removing the supernatant, digest the slurries by adding 10 milliliters of pre-warmed 0.2% collagenase type 11. Incubate for 60 minutes at 37 degrees Celsius, shaking the tubes by hand every 10 minutes. When the 60-minute incubation is done, centrifuge, and then resuspend the slurries in 10 milliliters of dispace solution. Incubate for 45 minutes at 37 degrees Celsius, shaking by hand every 10 minutes. After the 45-minute incubation, centrifuge and resuspend the slurries in 10 milliliters of 0.2% trypsin HBSS solution. We usually incubate for 15 to 20 minutes at 37 degrees Celsius while inverting the tubes every 10 minutes. The incubation is complete when enough single cells are released from the tissues, as illustrated here. Centrifuge the resulting cells and resuspend the cell pellet in 5 milliliters of proliferation medium, PM. Dissociate the cell suspension by passing the extract through a series of needles, 2 times through an 18-gauge needle, then once through a 23-gauge needle, and finally once through a 27-gauge needle. Lastly, pass the cell extract through a 70-micron cell strainer. To begin the pre-plating technique, centrifuge the cell extract that was just prepared and resuspend the pellets in proliferation medium. Plate 5 milliliters of the cell mixture onto a collagen-coated T25 flask and mark it as PP1. Incubate at 37 degrees Celsius in a humidified 5% carbon dioxide incubator for 2 hours. After 2 hours, transfer the non-adherent cells to a new collagen-coated T25 flask and mark it as PP2. Add 5 milliliters of PM into the previous T25 flask labeled PP1. Return both flasks to the incubator for 24 hours. After 24 hours, transfer non-adherent cells from PP2 to a new collagen-coated T25 flask and mark it as PP3. Add 5 milliliters of proliferation medium into the flask labeled PP2. Return the flasks to the incubator. Repeat the procedure every 24 hours until population PP6, the stem cell population, is created. The isolated PP6 cells are very few in number and need to be maintained in FBS-rich proliferation medium that is changed daily. Although most of the cells in the PP6 culture will die during the following 1 to 2 weeks of culturing, a large, healthy PP6 population is usually created after an additional 1 to 2 weeks of expansion. Maintain all other groups of suspended cells as well, allowing them to proliferate and examining them regularly using an inverted microscope. The early adhering cells that attach to the PP1 and PP2 flasks are mostly fibrotic cells. Cells that adhere to collagen-coated flasks after a longer time period within 48 to 96 hours, PP3 and PP4, are mostly myoblasts, while muscle satellite cells are often seen mostly in PP5. The later pre-plated cells, 96 hours or later, PP6, are regarded as adult stem cells and appear small, round, and translucent at the beginning of their isolation. They can be further characterized by immunohistochemistry for their expression of stem cell markers such as stem cell antigen 1 and CDC34.
We've just shown you how to isolate stem cell from a skeletal muscle of the mouse. When doing this procedure, it's important to remember to maintain sterility during all the procedures. Once the stem cell has been isolated, make sure to culture those cells at low densities to maintain the stem cell characteristics of the cells. So that's it. Thank you for watching and good luck with your experience.
And I wish you good luck to try to use this in your experiment. Thank you.